Sumateraland, an island on the west side of Indonesia. Its beauty and coastline spreads from north to the southern side. Inside of it, there is Bengkulu, a mesmerizing province with variety of biodiversity and natural resources. A home for unique and exotic flora and fauna which cannot be seen in anywhere else. Bengkulu was formed in 1968. With all the struggle, passion, and support, the obligation to provide a higher educational institution was made, the University of Bengkulu. As the biggest university in Bengkulu has an important role to strengthen the character, provide knowledge, and develop all existing potential that Bengkulu has. From the land of Asia, University of Bengkulu creates a better and knowledgeable human resources to compete in any level. We provide the facilities as best as we could to help you learn, research, and gain more experience in all of this rapid development. Our educators are continuously being improved so you may absorb the better understanding, the better knowledge to conquer the world. Education and research are our biggest missions to be improved. We always encourage all students and academic activities to produce their own work with copyright of intellectual properties. Implement the knowledge directly to the society, locally, and even internationally. And for sure, develop our system management. All of those are done to achieve one goal: become the world-class university. In this millennial era, we understand that education is not just a media to find knowledge, but also a huge container to form the better character and more competitive human that has a big desire to challenge themselves. From every corner of classes, we prepare and teach our students to get ready to face the world. The golden generation, with heart full of fiery desire and head full of sparkling knowledge. From every corner of classes, we pack our students not only with theory but experience, so they may serve themselves to their beloved country. University of Bengkulu is the best place for every knowledge seekers from every part of the world, a place to discuss and understand each other. All become one, blend in a harmony of diversity. University of Bengkulu is the best place to create work. Every idea counts. We merge all of those into one greatest output. In here, we may expand our mind and express ourselves freely. For us, it is very important to build a big and strong connection to every person, to give them the very best impression, so the rapid movement will not block our ways to reach success. University of Bengkulu is well known with its achievement, the proofs that stated our power and potentials. In here, we prepare ourselves to face the world, to build our future, to reach our dreams, and to welcome the expected goals. prepare the best supplies towards the stronger nations by sharpening our knowledge and experience. In here, in University of Bengkulu, from Bengkulu for Indonesia, reaching the world.
Saat ini, kita berada dalam masa sulit. Terbatas dalam gerak, terpaksa oleh jarak. Menjauh dari dunia yang sesak. Yang tak lama, tak sejenak. Saatnya berjeda. Menjaga jarak, satu-satunya solusi. Ada kalanya kita perlu berhenti agar semakin cepat pulih kembali. Perhentian ini pun menjadi sebuah momen untuk kita merefleksikan diri. Waktunya untuk peduli, memberi ruang untuk dunia sedikit beristirahat. Banyak rindu yang harus ditahan, banyak rencana yang harus ditunda. Sebarkan harapan untuk keselamatan bersama. Kita semua saling menguatkan. Ini adalah momen untuk kita mencari sudut pandang yang tak pernah kita temukan sebelumnya, menjadikannya sebagai energi untuk mampu bergerak kembali. Ini adalah momen untuk semua menyatukan asa dan harapan. Dan ketika hari itu tiba, semua ini akan membaik. Manusianya, begitupun semestanya.
Bengkulu, sekeping surga yang terpendam di balik rimbunnya tanah Sumatera. Bermacam keunikan beramu padu menjadikan Bengkulu berbeda dan tak dapat dijumpai dimanapun. Salah satu yang paling berpengaruh dalam perkembangan Bengkulu adalah pendidikan. Dan Universitas Bengkulu, ikon pendidikan tinggi di Bengkulu, bidang sains, sosial, dan teknologi. 8 fakultas yang tergabung di dalamnya, satu visi mewujudkan World Class University 2025. Fakultas Matematika dan Ilmu Pengetahuan Alam adalah salah satu yang paling gencar mewujudkan visi itu. Hal ini dibuktikan dengan fakultas yang leading dalam percepatan mencapai visi World Class University. Fakultas yang pernah dipandang sebelah mata ini kini dapat berbangga dan semakin percaya diri, bergerak cepat dan berinovasi demi menghadapi persaingan dalam bidang ilmu pengetahuan. Sejarah panjang pendirian FMIPA sejak 1992 merupakan perjuangan heroik yang kemudian menanamkan nilai-nilai mewujud menjadi etos kerja, sehingga FMIPA memacu diri untuk semakin memimpin.
Saya adalah salah satu pelaku sejarah berdirinya FMIPA. Memang sejak SMA saya sudah sangat ingin sekali mempelajari ilmu kimia. Oleh sebab itu, setelah tamat SMA, saya masuk di jurusan kimia FMIPA. Namun ada beberapa kendala yang dihadapi di tengah perjalanan yang menyebabkan memang saya harus berhenti kuliah, bahkan saya pindah jurusan. Pada akhirnya saya tetap menyelesaikan studi saya di jurusan Kimia FMIPA. Dan setelah itu saya melanjutkan S2 di UNAN dan S3 di UGM. Dan alhamdulillah sekarang saya sudah menjadi salah satu staf pengajar di jurusan Kimia FMIPA Universitas Bengkulu.
Dengan semangat dan kerja keras, perjalanan panjang yang diwarnai peluh dan rintangan, Fakultas Matematika dan Ilmu Pengetahuan Alam tetap konsisten dengan komitmennya. Surat keputusan dari Kementerian Pendidikan dan Kebudayaan 12 Agustus 1999 menjadi tonggak sejarah Fakultas MIPA. 2 tahun berselang, Fakultas MIPA pun menerima mahasiswa baru dengan membuka 4 program studi sekaligus, yaitu S1 Matematika, S1 Biologi, S1 Kimia, S1 Fisika, untuk menyelenggarakan pendidikan yang profesional, riset yang bermutu, dan menerapkannya untuk kesejahteraan masyarakat, Fakultas Matematika dan Ilmu Pengetahuan Alam Universitas Bengkulu terus berkembang dengan mendirikan berbagai program studi. D3 Lab Science, S2 Statistika, S1 Statistika, S2 Kimia, Adanya Poltekkes Provinsi Bengkulu dengan Universitas Bengkulu menjadi kekuatan baru bagi Fakultas Matematika dan Ilmu Pengetahuan Alam. Amanah penggabungan ini diterima FMIPA yang membuat prodi FMIPA semakin beragam. Prodi-prodi tersebut adalah D3 Keperawatan, D3 Kebidanan, D3 Farmasi, Ditambah lagi pada tahun 2017, Fakultas Matematika dan Ilmu Pengetahuan Alam resmi menerima surat keputusan tentang pendirian 2 program studi baru, yaitu S1 Geofisika, S1 Farmasi. Menyikapi tantangan yang ada, Fakultas MIPA Universitas Bengkulu telah menetapkan rencana strategis 9 tahun yang akan mengubah orientasi proses maupun output kepada peningkatan kualitas. Aktivitas riset dan publikasi ilmiah pada jurnal terakreditasi dan jurnal internasional menjadi indikator kunci dari kinerja dosen FMIPA UNIF. Demikian pula proses edukasi efisien, tepat waktu, terstandarisasi, serta efektif dalam menghasilkan lulusan yang kompeten menjadi parameter kunci atas kinerja manajemen fakultas dan kompetensi dosen maupun tenaga kependidikan. Dengan semakin besarnya aktivitas riset, maka tuntutan terhadap institusi fakultas untuk lebih berperan dalam pengelolaan riset semakin besar. Sumber daya yang digunakan harus efektif sehingga hasil penelitian memberikan manfaat maksimal tidak saja bagi penelitinya, namun juga bagi masyarakat luas. Dengan tenaga pengajar dan sumber daya manusia yang profesional serta didukung dengan sarana penunjang, Fakultas Matematika dan Ilmu Pengetahuan Alam siap menjadi lembaga pendidikan, penelitian, dan pengabdian bidang sains dan teknologi kelas Asia Tenggara tahun 2025.
Saat ini, kita berada dalam masa sulit. Terbatas dalam gerak, terpisah oleh jarak. Menjauh dari dunia yang sesak. Yang tak lama, tak sejenak. Saatnya berjeda. Menjaga jarak, satu-satunya solusi. Ada kalanya kita perlu berhenti agar semakin cepat pulih kembali. Perhentian ini pun menjadi sebuah momen untuk kita merefleksikan diri. Waktunya untuk peduli, memberi ruang untuk dunia sedikit beristirahat. Banyak rindu yang harus ditahan, banyak rencana yang harus ditunda. Sebarkan harapan untuk keselamatan bersama. Kita semua saling menguatkan. Ini adalah momen untuk kita mencari sudut pandang yang tak pernah kita temukan sebelumnya, menjadikannya sebagai energi untuk nanti bergerak kembali. Ini adalah momen untuk semua menyatukan asa dan harapan. Dan ketika hari itu tiba, semua ini akan membaik. Manusianya, begitupun semestanya.
Sumatra Land, an island on the west side of Indonesia. Its beauty and coastline spreads from north to the southern side. Inside of it, there is Bengkulu, a mesmerizing province with variety of biodiversity and natural resources. A home for unique and exotic flora and fauna which cannot be seen in anywhere else. Bengkulu was formed in 1968. With all the struggle, passion, and support, the obligation to provide a higher educational institution was made, the University of Bengkulu. As the biggest university in Bengkulu has an important role to strengthen the character, provide knowledge, and develop all existing potential that Bengkulu has. From the land of Asia, University of Bengkulu creates a better and knowledgeable human resources to compete in any level. We provide the facilities as best as we could to help you learn, research, and gain more experience in all of this rapid development. Our educators are continuously being improved so you may absorb the better understanding, the better knowledge to conquer the world. Education and research are our biggest missions to be improved. We always encourage all students and academic activities to produce their own work with copyright of intellectual properties. Implement the knowledge directly to the society, locally, and even internationally. And for sure, develop our system management. All of those are done to achieve one goal: become the world-class university. In this millennial era, we understand that education is not just a media to find knowledge, but also a huge container to form the better character and more competitive human that has a big desire to challenge themselves. From every corner of classes, we prepare and teach our students to get ready to face the world. The golden generation, with heart full of fiery desire and head full of sparkling knowledge. From every corner of classes, we pack our students not only with theory but experience, so they may serve themselves to their beloved country. University of Bengkulu is the best place for every knowledge seekers from every part of the world, a place to discuss and understand each other. All become one, blend in a harmony of diversity. University of Bengkulu is the best place to create work. Every idea counts. We merge all of those into one greatest output. In here, we may expand our mind and express ourselves freely. For us, it is very important to build a big and strong connection to every person, to give them the very best impression, so the rapid movement will not block our ways to reach success. University of Bengkulu is well known with its achievement, the proofs that stated our power and potentials. In here, we prepare ourselves to face the world, to build our future, to reach our dreams, and to welcome the expected goals. We prepare the best supplies towards the stronger nations by sharpening our knowledge and experience. In here, in University of Bengkulu, from Bengkulu for Indonesia, reaching the world.
Saat ini, kita berada dalam masa sulit. Terbatas dalam gerak, terpaksa oleh jarak. Menjauh dari dunia yang sesak. Yang tak lama, tak sejenak. Saatnya berjeda. Menjaga jarak, satu-satunya solusi. Ada kalanya kita perlu berhenti agar semakin cepat pulih kembali. Perhentian ini pun menjadi sebuah momen untuk kita merefleksikan diri. Waktunya untuk peduli, memberi ruang untuk dunia sedikit beristirahat. Banyak rindu yang harus ditahan, banyak rencana yang harus ditunda. Sebarkan harapan untuk keselamatan bersama. Kita semua saling menguatkan. Ini adalah momen untuk kita mencari sudut pandang yang tak pernah kita temukan sebelumnya, menjadikannya sebagai energi untuk nanti bergerak kembali. Ini adalah momen untuk semua menyatukan asa dan harapan. Dan ketika hari itu tiba, semua ini akan membaik. Manusianya, begitupun semestanya. Saat ini, kita berada dalam masa sulit. Terbatas dalam gerak, terpaksa oleh jarak. Menjauh dari dunia yang sesak. Yang tak lama, tak sejenak. Saatnya berjeda. Menjaga jarak, satu-satunya solusi. Ada kalanya kita perlu berhenti agar semakin cepat pulih kembali. Perhentian ini pun menjadi sebuah momen untuk kita merefleksikan diri. Waktunya untuk peduli, memberi ruang untuk dunia sedikit beristirahat. Banyak rindu yang harus ditahan, banyak rencana yang harus ditunda. Sebarkan harapan untuk keselamatan bersama. Kita semua saling menguatkan. Ini adalah momen untuk kita mencari sudut pandang yang tak pernah kita temukan sebelumnya, menjadikannya sebagai energi untuk mampu bergerak kembali. Ini adalah momen untuk semua menyatukan asa dan harapan. Dan ketika hari itu tiba, semua ini akan membaik. Manusianya, begitupun semestanya.
Sumatra Land, an island on the west side of Indonesia. Its beauty and coastline spreads from north to the southern side. Inside of it, there is Bengkulu, a mesmerizing province with variety of biodiversity and natural resources. A home for unique and exotic flora and fauna which cannot be seen in anywhere else. Bengkulu was formed in 1968. With all the struggle, passion, and support, the obligation to provide a higher educational institution was made, the University of Bengkulu. As the biggest university in Bengkulu has an important role to strengthen the character, provide knowledge, and develop all existing potential that Bengkulu has. From the land of Asia, University of Bengkulu creates a better and knowledgeable human resources to compete in any level. We provide the facilities as best as we could to help you learn, research, and gain more experience in all of this rapid development. Our educators are continuously being improved so you may absorb the better understanding, the better knowledge to conquer the world. Education and research are our biggest missions to be improved. We always encourage all students and academic activities to produce their own work with copyright of intellectual properties. knowledge directly to the society, locally, and even internationally. And for sure, develop our system management. All of those are done to achieve one goal: become the world-class university. In this millennial era, we understand that education is not just a media to find knowledge, but also a huge container to form the better character and more competitive human that has a big desire to challenge themselves. From every corner of classes, we prepare and teach our students to get ready to face the world. The golden generation, with heart full of fiery desire and head full of sparkling knowledge. From every corner of classes, we pack our students not only with theory but experience, so they may serve themselves to their beloved country. University of Bengkulu is the best place for every knowledge seekers from every part of the world, a place to discuss and understand each other. All become one, blend in the harmony of diversity. University of Bengkulu is the best place to create work. Every idea counts. We merge all of those into one greatest output. In here, we may expand our mind and express ourselves freely. For us, it is very important to build a big and strong connection to every person, to give them the very best impression, so the rapid movement will not block our ways to reach success. University of Bengkulu is well known with its achievement, the proofs that stated our power and potentials. In here, we prepare ourselves to face the world, to build our future, to reach
Yes, almost at 9 o'clock, right? 9 o'clock evening.
No, it's only 7 o'clock,
Oh,
7 p.m.
oh, okay.
Can I share my screen yet, or should I wait?
Yes, maybe we have to wait for several minutes.
Okay.
USA time. Are you hearing to me?
7 p.m. early evening.
7 p.m. evening, yeah. Okay, so how are you doing today?
We are doing very well. Thank you.
Yeah,
And you?
yeah, we still have time waiting for another keynote speaker.
Okay.
Yeah, yeah, okay, we just wait. Thank you, thank you.
Assalamualaikum warahmatullahi wabarakatuh. And good morning, ladies and gentlemen. Good morning to all of the participants and also the presenter who has been joined in this panel. Yes, and also good evening for Mr. Wali Fansikal in USA. Okay, before we starting for our presentations, for our both of keynote speaker by Mr. Wali Pansikal and also Mrs. Dr. Sastya Pramaputri from Osaka University. But before that, maybe there are some of the warm greeting from our chairman for the second days to Dr. Rizwar. Time is yours.
Thank you. Assalamualaikum warahmatullahi wabarakatuh. Hello, good morning to all in these conferences. This morning, we still can join in plenary session second day. We are so happy because our distinguished keynote speakers, Wali Pansikal PhD and Dr. Satya Pramaputri MSc have been joined with us in this panel on second day of the third KOBI Congress and International and National Biologi conferences in Bengkulu. And also join with us the invited speakers, Professor Dr. Antonio Suwanto MSc, Professor Dr. Zainal Muktamar MSc, Professor Dr. Ani Mariastuti MSc, Professor Dr. Pinkan Adiwati MSc, and Dr. Bambang Retnoaji MSc. Dear ladies and gentlemen, Alhamdulillah, on the second day of this conference, we hope that everyone is is good in health and still excited to join in this event. Yesterday, two previous keynote speakers have talked about the current and future Indonesian biodiversity conservation policies and challenges by Insinyur Wiratno MSc, and the contribution of KOBI in supporting Indonesian biodiversity by Professor Dr. Budi Setiadi Daryono, Master Agriculture Science. And today, we still have two other keynote speakers from the USA and Japan, namely Wali Pansikal, PhD, with the title Biological Research Technology and Technology Research in the Field of Conservation Biology. Dr. Satya Pramaputri, MSc, with the title The Role of Metabolomics to Support Indonesian Biodiversity Conservation. After that, 5 of invited speakers will start a presentation according to their respective field of expertise in the parallel session. We hope that this international conference will benefit to all of us and contribute to accelerate Indonesian development, especially in the progress of science and technology, which is currently entering the era of the Industrial Revolution 4.0. Finally, happy holding the international conference. I hope you will be happy with this activity. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you to our chairman of the committee, Dr. Rizwan. And then now we still continue for our agenda for today. So we are very happy here, me, Rizky Hadibo, and also my partner,
Khairul Muslim.
So now we still back again in the second session. So we are from the Department of Biology, Faculty of Mathematics and Natural Sciences, University of Bengkulu. So will be your moderator for this panel session. Okay, so we are very glad to meet you, all of the participants here. So, and then we try to continue for our first presentations. Okay. But before we start for the presentations from Mr. Wali Pansikal, maybe we try to read the curriculum vitae or bio from Mr. Wali. Okay. Okay, Mr. Wali Pansikal is the president of Idea Wild. He graduated from Geology and Mineral Chemistry, South Dakota State University, Brookings. His master at Geology and Physiology, University of Wyoming, Laramie, and his research topics include conservation biology, geology, and physiology. He was a research biologist at Rocky Mountain Forest and Range Experiment Station, American Martin Research Project in the Sierra Mountains, immobilize and radio-colored martins, monitor martin movement with radio telemetry equipment, Smithsonian Conservation and Research Center in US, Migrant Bird Research Project in Belize, and many other research. He was a wildlife biologist at Green Plain Wildlife Institute. He also was a biology teaching assistant at University of Wyoming, prepared and presented lecture, design, and administered assignment, demonstrate and supervise lab technique, tutor, receive outstanding general biology teaching award. Okay, maybe now we entering for the sessions of the presentations from Mr. Wali. I think, Mr. You can share screen for your PowerPoint.
Time is 30 minutes. Yeah. Please, Dr. Walter Dean. Okay, time is yours, Mr. Walter Dean.
Okay. We need to wait 1 minute for this thing to load and we can go. Can you hear me okay?
Yes, we can hear you. Is it
Okay.
clear?
course.
The,
Voice is clear.
the program is loading. We just have to wait about a minute. While we're waiting, I just want to say it's an honor to be here. Tonight, I want to give a bit of a big picture, tell you about a nonprofit that I created over the years, show you some projects that we've helped out with, some conservation projects, show how to apply for equipment. I think some of you are interested maybe in equipment in the future to apply for. And in the meantime, I wanted to basically, before I get into the nonprofit organization, I want to tell you a little bit about, I'd like to share a dream. I'd like to share what I'm passionate about, and the tools that fuel that passion, because I think they're common to all of us. And so these are things that tools all of us can use in the future to help us with our conservation efforts. I normally, if I do this presentation live, I would ask people in the audience what they are passionate about. And I know people like Dr. Wilson Navarino are passionate about tapirs and passionate about bird watching. And I know my friend Gunawan is passionate about helmeted hornbills. And I know my friend Dr. Puan is passionate about owls. I am very passionate about, and I know many of the people in the audience are passionate about cooking or hiking or travel or music or many other things. Okay. I want to tell you a little bit about what I'm passionate about. Back in 1971, when this photo was taken, I was extremely passionate about dinosaurs. And I was so passionate, I created a dinosaur club. And I didn't have many members, just my friend Brett Jeske on the left and my little brother on the right, but we had a dinosaur club. And because of that passion, I later became interested in the field of zoology. And I went on to get a degree in zoology. And after that degree, I had a couple choices to make. I had a degree in zoology, but I did not know what to do with it. So the theme of this presentation is we're going to see that the helpful tools we need to accomplish our missions and our passion are good mentors. We need to do some wandering, and we need to volunteer. So I went off to Kenya, East Africa, and I volunteered at a veterinary clinic with Dr. Barry Cocker. And for 2 weeks, I worked with this wonderful individual. And after 2 weeks, I decided I did not want to be a veterinarian. So we became very good friends, but I learned that from this volunteer experience, I wanted to become something else. I spent considerable time bird watching with this lady in Kenya, Flor Ngwenu, who is very passionate about birds. And she introduced me to people doing conservation projects all over Kenya. And one of those people worked at Olagasali Prehistoric Site. His name was Leon Bennett. And he took me on for 2 months, and I volunteered with him, and I got to work with, lo and behold, little tiny dinosaurs. I love dinosaurs. I wanted to work with dinosaurs, and I ended up getting to work with little tiny dinosaurs in Kenya. And you'll notice some of these birds are familiar: barbets, sunbirds, hornbills. And while I was working with Leon later became the head of BirdLife International, so he was a great mentor. But while he was mentoring me, I noticed he had really poor equipment. His mist nets were so bad that they had holes in them, and a lot of the times the birds escaped. And so I made a mental note of that. And the rest of the time I spent in Kenya, I just did some wandering. And so I had watched many television shows about all the species found in Africa. And so I went out and I got to see elephants in the wild, and I got to see zebras in the wild, and many, many different species of birds like these oxpeckers, impalas, elands. This was an antelope I'd never even heard of. Of course, we got to see all the major predators, lions and more lions, cheetahs. And because of this experience and wandering around East Africa, I really fell in love with our natural world. I already was, but this really tightened the bond, and I really knew I wanted to play a role in conservation and wildlife conservation. I even got to meet animals I'd never heard of, like banded mongooses. And an interesting side note is 20 years after my trip to Kenya the first time, I showed up and I was giving a presentation for Idea Wild at the Natural History Museum. And Flora Ngueno, that same lady who was the passionate bird watcher, she walked in and watched the presentation. She was still conducting bird walks in Nairobi, Kenya. And as far as I know, she still is. And she's almost 90 years old now. So this lady has a lot of passion in her eyes, and she inspired many people. And she was one of my many mentors in Kenya. So after I knew what I wanted to do in my career, I wanted to become a wildlife biologist. I once again relied on good mentors, wander, and volunteer. And so I bought a car and I drove around the western side of the United States. And I was looking for a good professor I wanted to work with. And I met this man down here, Dr. Fred Lindsay. And I met this cowboy over here who had worked with mountain lions since he'd been a very young boy. And so those two became my mentors in southern Utah. And for the next 2 years, I studied mountain lions for my master's degree. And we captured these animals using dogs up in trees. We put radio collars on the animals. We tracked them using radio telemetry. And I learned what it was like to become a wildlife biologist. And just this shot, I like this shot because it's my father. And I just want to say our mentors often come in many, many forms. And our mothers and fathers and friends are oftentimes very good mentors as well. So on this project, I had everything I could dream of. I had helicopters. I had other kinds of helicopters. I had airplanes. I had trucks, horses, snowmobiles, technicians to help do the work. Everything a biologist could dream of. And I just made a note of that, and I want you to remember that on this project, that's what I thought was normal in all other projects. So after those 2 years, I became a wildlife biologist, and I got a master's degree in zoology. At that point, myself and several friends were very tired of being students, and we needed to go out and wander again. So we packed up our backpacks and we flew off to Australia. And there we found good mentors. We volunteered and we did some more wandering. So we packed up, we bought a car there. We packed it up full of camping gear. And once again, we spent almost 6 months chasing small dinosaurs. We went all over Australia, learning all the different species of birds and seeing all the different species of birds in forests, in coastal areas, in tropical wetlands, swamps. We've bird watched all over the place. And we bird watched up in canyons via canoe. And one of the things we learned from that is that wandering, that physical wandering, we learned many, many things. But we also did mental wandering, where we kind of stepped outside our normal belief system, because our beliefs are just kind of reoccurring patterns of thought. We just keep thinking the same thing a lot of the time. And so by wandering around, a lot of times we get to be on the other side of our limited beliefs. So I love the freedom that exists on the other side of limited beliefs. And I think wandering is really helpful for our passion. And another thing, while we were there, we volunteered with Dr. Todd Soderquist, who was working with small mammals to conserve small mammals in Australia. And this was the species he worked with, and they were nocturnal. And one night, the equipment he was using, which was a spotlight, a little handheld spotlight, it stopped working, and he was very upset about that. And I didn't understand why. And he said, I cannot afford a $40 spotlight. And for me, coming from projects where I had helicopters and airplanes and all these things, it made no sense to me that a conservation biologist on the front lines of conservation would not have basic equipment. So he recommended one more wandering trip for us. We went out to the Great Barrier Reef, and we spent 1 week out there snorkeling and getting in touch with the marine animals that live there, and seeing animals and species that I'd never seen before in my life, like triggerfish and coral and all the different things, these giant clams, spotted eagle rays. We watched baby turtles hatched, got to swim with sea snakes. And these were all experiences I'd never had before. So once again, I fell in love with the ocean and all the life forms on this planet because we had spent some time wandering around with them. Now, the second week we were on this island, a hurricane hit or a cyclone hit, and it knocked down trees on the island, and our boat could not come get us. So we spent 1 week in this tent waiting for the wind to go down. And during that time, I came up with the idea to start a nonprofit that helped other people with the equipment they need, the tools they need to do conservation projects. So because we spent some time wandering around Australia, this idea came to me. We even had to hunt for our own food. And after 2 weeks, the boat finally came and got us. So I came back to the United States. I decided I was going to start a nonprofit organization, but a friend of mine asked me to join him on one more volunteer experience. So once again, good mentors, wander and volunteer. I was asked to come to Belize, where I got to work with these two people on the left, Patricio, and on the right, Courtney Conway. Now, Patricio became one of my best mentors ever. He was an indigenous Mayan Indian, and he knew everything about tropical rainforests. And he introduced me to the life that's found in tropical rainforests that I would never have seen before or known about. And my good friend Courtney Conway was an amazing ornithologist that taught me how to study birds and how to collect data and the equipment you need to study birds. So we spent basically, once again, 3 months chasing small dinosaurs and collecting information on them and how they lived in primary forests, secondary forest, in areas that have been totally destroyed. Some of these species never seen before. This is a hummingbird. This is a flycatcher, manakin, tanager, mott, type of woodpecker. And we also got to learn what it was like to see the jungle or the rainforest through the eyes of an indigenous group, because we spent every night living in their houses. So there's one more tip I would like to add before we get to the nonprofit organization that has been very helpful for me, and that's the ability to visualize or use your imagination. Oftentimes in conservation, we get stuck and we think, oh, I can't do that, or I can't study that bird, or I can't work in that community, or I can't raise enough money. We need to put ourselves and imagine ourselves in situations where all those things are happening. And we need to imagine ourselves actually and feel ourselves in those situations, and we will become much more successful. When you are in a sporting event like the Olympics and you're a gymnast and you're about to do your routine, you visualize your entire routine before you go out and do that routine. When you're in track and you're about to run across the finish line, you visualize yourself running across the finish line. We can do the same thing in conservation. Right now, many of us are faced with enormous challenges in conservation, and we need to visualize how we're going to get through that and how we're going to be successful at it. It's a very important tool. Many people teach it. I learned some lessons from Paramahansa Yogananda, from Dr. Joe Dispenza, from Esther and Jerry Hicks. There's a lot of sources to learn about visualization. And if you want a great movie, this movie can be rented on Netflix, I believe, or Amazon. What the Bleep Do We Know? It actually talks about the physics of visualization and using your imagination. This is something we were born with as children, and we really need to take advantage of our imagination and use visualization. Okay? So that very thing, that wandering, that mentorship, that volunteering, and that visualization led to the creation of a nonprofit organization called Idea Wild, which I've been working at now for 30 years. And like we started it back in 1991, and the idea was to help conserve the biodiversity of the planet. We all know what biodiversity is. So the mission statement is to equip, empower, and activate the world's most promising environmental leaders to grow and strengthen the movement to conserve the planet's biodiversity. We all know the importance of biodiversity. We use it for food, fiber, pharmaceuticals, healthy ecosystems, pollination. It's the underlying feature of our entire economic system. If we had no ecosystems, the whole thing would collapse. For example, this pool in Yellowstone National Park, which is under conservation efforts in the United States, holds little tiny microbes called Thermus aquaticus. You can pull an enzyme out of them, and that's what most of our genetic research we're able to do today is because of an enzyme found in that national park. This plant grows in Madagascar, rosy periwinkle. If you grind it up, you can make one of the top 5 cancer drugs. So we know this biodiversity is very important. We also know there are heroes and sheroes that are working to conserve this biodiversity, like this very famous person, Jane Goodall. And this wonderful person, Steve Irwin, who even got people to appreciate snakes. He left us too early, but his daughter is taking over where he left off. The world right now very much needs environmental leaders. We need political leaders, we need religious leaders, we need business leaders, we need sports leaders, we need teachers and leaders, but what we also very much need is environmental leaders. And I hope many of you, the people in the crowd, will become, some of those already are, and will become future environmental leaders. We like to call them the heroes and sheroes of the planet. So when we started Idea Wild, we worked in Mexico and Guatemala, and we looked for those heroes and sheroes of the planet. And we found them in professors, and we found them in their students working to conserve the biodiversity of their countries. And in most cases, those people did not have the equipment they needed to do their jobs. So these are some examples of projects we helped in different countries, in Bolivia, Brazil, the Caribbean, Colombia, Costa Rica, with birds, dolphins, butterflies, sea turtles. And you can see the type of equipment that was needed. Basic tools, GPS, binoculars, cameras, mist nets. Here's one in Indonesia, spotting scope, Madagascar, laptop, wolf research, video camera. So you can see all these types of projects, any kind of biological diversity, and any biologist, whether they're working on a bachelor's, a master's, a PhD, a postdoc, or working for a conservation organization. We're looking for people who are passionate about conserving biodiversity. I want to show you a few examples so you get a better idea. This project is in Thailand. Her name is Tan for short. And Tan went through the National Institute of the Royal Institute of Forestry there. She ended up getting her PhD in the United States. And she's looking at the corridor between Myanmar and Thailand and helping to conserve that area. It sits between 2 large protected areas in Thailand. She's looking at how hornbills move in this corridor. She works with the wreathed hornbill, and she works with the giant hornbill. And those 2 species help her discover where this corridor is. She's mapping all their nesting areas. All the nest holes are being mapped, as well as the feces is collected so she can determine what species they're dispersing. And this picture shows her, these are all the mapped nest sites she's found so far. On the Thailand side, this is the Myanmar side. So she's working on this side to provide a corridor so that those species can continue to exist. Idea Wild provided her with a spotting scope. We provided her with a GPS system, and we provided her with some other basic tools to do research on that bird. This project is with another bird species in Costa Rica with scarlet macaws. 15, 20 years ago, there was only 300 of these animals left in Costa Rica. And then this man decided he wanted to do something about that. So he asked Idea Wild for climbing equipment. We provided him with climbing ropes, and he goes up into the canopy and he attaches artificial nest boxes in areas that are secured from poachers. And now that population of birds has risen to 600 birds. And even the president of Costa Rica went up on these ropes to unveil the first nest box on national television. So some of these projects get very high profile. This is another project in Costa Rica with resplendent quetzal, considered to be one of the most beautiful birds in the world. We knew nothing about it. This young Costa Rican biologist was working on his master's degree. We basically only helped nationals, so only people from those countries. He needed binoculars, plant tagging equipment, and a GPS system. We provided that equipment for him. He studied that bird for 2 years, turned the research over to the Costa Rican government, and he created the newest national park in Costa Rica, named after the bird, Parque Nacional Los Quetzales. So incredibly, he was only 24 years old when he did this. This last project I will show you is in the country of Zimbabwe, and it works with different species of vultures. Most of you know there's a crisis with vultures in Asia and a crisis with vultures in Africa. This is the study area, which is magnificent. Lots of high cliffs, high profile topography. And this is one of the other species, lappet-faced vulture he works with. And anyway, in order to investigate nesting sites and look at the landscape, look at nesting trees, we provided him with a drone, a laptop, and some other basic equipment. So by now you understand we are specializing in providing equipment to people on the front lines of conservation. Here you see Michelle, who works to generate maps of different vegetation in Botswana. She needed a laptop computer. Anna Horvath works in Mexico with small mammals, needed small live traps. David Valenzuela needed tomahawk live traps. Javier Beltran needed a used motorcycle, telemetry equipment, binoculars, Write in the Rain notebooks, headlamps, equipment to work at night, Pensola scales, and even equipment for conservation education, like puppets or coloring books or video cameras or LCD projectors, things like that. Luis Sigler worked in Mexico with crocodiles. He had a boat, but he did not have a motor, so we donated a motor. Spotting scopes, camping equipment like backpacks and tents, and of course, GPS systems. And all these heroes and sheroes of the planet, or these future environmental leaders, are working on masters and PhDs and bachelor's degrees, and they're creating all sorts of data, and they're creating videos, coloring books for children, guidebooks. So they're creating all kinds of information we can use now to conserve biodiversity. And in the long run, we're also helping create experience and expertise, because every single one of these people has more experience now and more expertise because they've been out in the field using equipment. So both short-term and long-term conservation. Now, if any of you are interested in working to conserve biodiversity in the future or now, the application form for Idea Wild is on our website. We need some various pieces of information, your email address. We need a person in the United States who can receive the equipment and help you obtain that equipment. These are pretty self-explanatory questions. You'll have to fill out what type of equipment you want on this requested equipment form. So the name of the equipment, the item, the price, the quantity, total price. You can ask for equipment that goes all the way up to $1,500. There's a lot of equipment in that range. We're able to get computers for around $200. We're able to get camera traps for only $60 a piece. We're able to get mist nets for about $50 a piece. We work with a lot of suppliers, so we're able to get a lot of equipment for a smaller amount of money. You can see the guidelines on our website. You can review that. We want to see an Idea Wild application form if you apply. A proposal, it can be in your native language. It can be in Bahasa Indonesian. A one-page summary of your project in English, your resume or CV, and a letter of recommendation from your advisor if you're a student. So you can email those to us. We'll review those proposals, and hopefully we can help you with your different conservation projects. Currently, Idea Wild is providing equipment to 40 to 50 projects every single month. And we've done that all the way through this pandemic. and I don't see us stopping anytime in the near future. So we're going to continue to do that. Since we began 30 years ago, we provide equipment directly to 6,850 conservation biologists. On average, every one of those individuals shares it with at least 2 colleagues. So we're estimating something like 20,000 projects have used equipment provided by IdeaWild. For a complete list of projects, you can check out ideawild.org. Please also join us on Facebook, and you can see updates from the field. You'll get information that's coming in from different projects from time to time, and you can see different people working on conservation all over the planet. So please join us on Facebook. I like to leave with this quote. I think this quote is really important. Sometimes we think we're small and we can't affect change, and yet that's not the truth. Do not underestimate the power of the small, the ability of an individual to go out in the world and make a difference. We often equate largeness with power rather than seeing largesse is in fact a limitation to change. Beautiful quote. All right. So in finale, I just want to say, please, everybody, use some helpful tools, find some good mentors, get out and do some wandering in the world, volunteer your time when you can, visualize and imagine how your future can be and how you're going to affect the future of conservation, and put your passion into action and get out in the world and follow your dinosaur. All right. So thank you, everybody. That was my presentation. I'll be happy to answer questions, I believe, when we get to the group question period, or if we answer them now, I'm not sure. That's up to the organizers.
Yeah. Thank you, Dr. Walter Dean and Sikel. It is very impressed that you show us your real life experience in the field that make the biology so live and so impressed in the field, and you have come to most of all over the world, so that this kind of experience will be useful for us, for audience. But please hold on, Dr. Walter Van Dien. Excuse me, Dr. Walter Dean, that please hold on, since we have to go through the second speaker, Ibu Sastia, so that after Bu Sastia have a presentation for next 30 minutes, then I will let you, Dr. Walter Dean and Dr. Sastia, to answer the question from the audience. And I remind all of the audience to write the question or impression in the chat room or chat space that available in the Zoom facilities. So let me say hello to Dr. Sastia. Good morning, Dr. Sastia.
Hello, good morning. From Osaka, Japan.
So it is at noon, almost noon at Osaka. Is that true?
Yes, it's true. It's 11:30 here in Japan.
We are now at 9:30.
Yes.
Okay, so we have a speaker from all over the world, that is from USA, and you from Osaka, Japan. So,
Yes, it's really great to be here today.
okay, thank you. Before I let you to have your speech, let me read your academic background in the curriculum vitae. Ibu Dr. Sastia Pramaputri is adjunct professor at IBB University, an assistant professor and She got her tenure at Graduate School of Engineering, Osaka University, adjunct lecturer at School Life Sciences and Technology ITB. Dr. Sasya Prama graduated from Bandung Institute of Technology, master at Osaka University in the field of biotechnology, natural product chemistry. She received her PhD from Osaka University at 2010, and in 2020, her research experience on GSPS Kakeni as a principal investigator and co-investigator. She also followed professor training on BPSRC in the British government. That is a Japan Partnering Award as project partner. She has authored 7 books, 40 journal publication, 9 articles in international media or society-related publication, and her research topic include metabolomics and microbiology. Dr. Sasya Pramaputri also serving as a reviewer and editor journals. She is a member of International Indonesian Scholar Association as a Secretary General, Formin Institute, this Indonesian Young Scientist Forum, as Managing Director and Society for Biotechnology Japan. She has involved in various projects related to metabolomic and microbiology. She has also developed various international and awarded on Miyata Award for early career at faculty member in engineering in 2020. And President Award Metabolomic Society on 2020, and many other achievements. So it comes to Dr. Saskia to have your speech. Time is yours.
Thank you very much for your kind introduction. I'm really happy to be here, and I hope that when the pandemic is finished, I could visit Bengkulu again because Bengkulu is a very special city because it's my mother's hometown.
Last year you have visited to Bengkulu, yeah, Dr. Saskia?
Yes. Yes, I was sent under the Dikti's mission for the world-class Symposium Cendekia Kelas Dunia from Kemenristek Dikti.
Association of Diaspora.
I was sent there as a diaspora to visit University of Bengkulu, and it was really a nice experience to go back with my mother. And I was really hoping and excited to be here, to be there again this year. But unfortunately, due to the pandemic, I have to deliver my presentation online. But I hope it still feels like through this online, we can still feel close to each other, and then we can share some useful ideas through this conference. So thank you so much for this. Okay, so may I start my presentation? You can see my slides and my voice clearly.
Start for your presentations.
Okay. So a very good morning, everyone. Assalamualaikum warahmatullahi wabarakatuh. So today I would like to talk about the role of metabolomics to support Indonesian biodiversity conservation. So although I've been working in Japan as a permanent faculty position here, and I've been here for 15 years, most of my research and the majority of my research group members are from Indonesia, in fact. And I still don't forget my hometown and consider that how to apply metabolomics technology for the conservation of Indonesian biodiversity. So as we all know, Indonesia is a country with very rich bioresource and abundant food production, but we also know that crop loss often occurs. And the main issues related in major crop loss in Indonesia are contributed due to, for example, disease in plant or agricultural product, poor post-harvest management, and also sometimes low quality of our produce. And how metabolomics can tackle these issues is through many different ways. So one of the ways is to investigate the cause of disease, and we could also aid systematic and effective post-harvest management strategy, and also how to improve the quality of Indonesian bioproducts. So for those who are not familiar with metabolomics, I would like to give you a little bit of introduction on what metabolomics is and the various applications of metabolomics. So you may know that in the central dogma of molecular biology, we know that the metabolites are the end product and the result of gene expression. So we start with the genome, and then afterwards we have the transcription and then translation, finally resulting in the phenotype. So before the phenotype, we have metabolism and then the involvement of many different kinds of metabolites involved in our metabolism. So this metabolism metabolic process is present in all living beings, which means from a very simple microbe to our own body as humans, we have metabolites all over, regulating many different functions in our body. As such, if we apply metabolomics technology, as long as they are living being and they have a metabolic process to regulate all of the cell functions, then we could do metabolomics using this organism. So the many different kinds of applications of metabolomics include, for example, to unravel gene function and how it interacts with bacterial, plant, animal, and human research. And this one is particularly very useful to understand the mechanism of plant diseases, for example, or diseases in our aquaculture product, for example, shrimp aquaculture, and then find out the ways to inhibit this pathogen infection or make the plants or our aquaculture product more resistant towards this disease. And also we can utilize our very rich bioresource to have more added value. It's not only selling it as a raw material, but we have to move to a bio-based economy where we could make use of our natural bioresource and then have a product that is more added value. So this is through the bio industry. Some of the examples are through the use of microbial hosts for example, production of various biochemicals, or we could utilize our rich bioresources and then utilize them in bioindustry to process them into products with higher value. And of course, the application of metabolomics originally are mostly concentrated on the application for medical research. For example, it can also be applied to COVID-19 research, in which we can find, for example, potential biomarker or metabolite biomarker in the patients that are more prone to getting severe infection. What kind of people with what kind of metabolite marker have, for example, the highest risk to get severe disease for COVID-19, for example. And therefore, then we can develop a very good diagnostic to protect this high-risk infection patients. And what I would like to talk about today is mainly focus on the application of metabolomics for food and agriculture for the purpose of quality improvement, to assist breeders in strategic trait optimization, or for post-harvest management. So to do my research group collaborates with many different kinds of international institutions. Mostly for food and agriculture, I focus only to collaborate with Indonesian entities. So at present, we have many collaborations with IPB University, with ITB Bandung, with industry, for example, with Biopharma and the chocolate company Pipiltin. We have a long-term collaboration also with Indonesian Coffee and Cocoa Research Institute. And we are now establishing a preliminary collaboration with the University of Bengkulu. So the concept of food metabolomics is as follows. So as long as you have any kind of food samples, you have to have a quantitative way to measure the quality. So for example, the quantitative way to measure quality can be by quantitative analysis of food function. It can be taste, flavor, grade, but it has to be measured quantitatively, not subjectively and not qualitatively. Once you have a quantitative value to measure the quality, then you could subject the same samples and subject them to analysis by different kinds of instruments. In our laboratory, we mainly use mass spectrometry, so chromatography combined with mass spectrometry to measure many different kinds of components in a biological samples at once. So instead of, please consider different ways of thinking. We don't just simply analyze, for example, 1 or 2 peaks in one biological sample. For example, targeting only on alanine or targeting only on glucose or targeting only on organic acid, maybe 1 or 2 organic acid. We measure hundreds of metabolites in a single run, and that can be done in 20 minutes. So within 20 minutes, you get the information of hundreds of peaks. And from among the hundreds, usually we can identify or annotate about 60 to 100 in one biological sample. Using the total component information and then the food function, you could correlate that with statistical correlation. So in here, you need mathematics. Then using this kind of correlation analysis, you can determine the correlation and the relationship between food function and food component. In short, for example, you can ask them the question, what makes your food delicious? What makes this food have higher quality? For example, like Kobe beef compared to other beef. What makes Kobe beef, for example, very tender, very delicious? Then what kind of component is high only in Kobe beef and not found in other food? So the way we can apply metabolomics can be many different ways. One application is just to tell the difference between one class of product and another class of product. For this, you can do discriminative analysis. Then you can get different biomarker to indicate the characteristic of a particular food group. Then you can also do that for informative. For example, like I mentioned, to assist breeders in optimizing their post-harvest management strategy, to understand what kind of mechanistic pathogen mechanism and how to understand the plant ripening process so that you could prevent ripening. These kind of things you can do the informative. And another one, you can also do it in a predictive way, means you can predict quality without the use of sensory analysis. So once a model is established between, for example, food quality and food metabolite profile, then you could use metabolite profile to predict the food quality without the need to do the sensory analysis. This would be very helpful because at present, if we use human as a sensory evaluator, sometimes it's subjective, and also you need a lot of training to get a very high, precise, good quality of the data of sensory. And also, our tongue is limited. You cannot analyze, for example, 100 coffee samples in one day. Maybe it's limited to, like, in one analysis, you could only analyze about 6 or 7 samples. Otherwise, we will have a fatigue. So to complement human sensory analysis, it's very useful if we have a complementary method that can be done in a very high throughput way, such as metabolomics. So I would like to now present to you an example. So this is the first time I present about this topic to Indonesian audience. So this is in preparation for submission, so very new. And how we can use metabolomics to propose quality marker for specialty coffee. I'm not sure if everyone is familiar with the specialty coffee. So whenever you drink coffee, probably some of you who are coffee lovers care so much about the quality. So if you are aware of the cupping test, so coffee quality is determined by cupping test, and it's usually done by a trained panelist, licensed Q grader, we say, for Arabica coffee. And the method is done using the Specialty Coffee Association of America method. So there's maximum scale is 100, from 0 to 100, the score. And if you get more than 80 points, it is categorized as specialty coffee. How to get 100? So there are 10 attributes that is shown here in the slide, and each attribute will get a scale from 0 to 10. So cumulatively, it will result in 100 as a maximum score. So this specialty coffee gained a lot of attention in recent years. If you are in the audience, I will ask you what kind of specialty coffee you like. So Indonesia is a country with the most variety of specialty coffee. We have Aceh Gayo, we have Mandailing coffee, we have Toraja, we have Java coffee, we have Bali coffee. We have coffee from all over Indonesia, and maybe about a dozens of specialty coffee from different origin, and they are all different. So I would like to propose a quality marker of specialty coffee by regression model-based metabolomics analysis. So the way to do that is we subject these specialty coffee, various kinds of specialty coffee, subject them to cupping tests, also component profiling at once, and then construct a model to see what kind of metabolite is always correlated with high quality coffee. So we have 10 different kinds of Indonesian high specialty coffee. We received these samples from the Indonesian Coffee and Cocoa Research Institute through one of the contests for specialty coffee in Indonesia. So every year we have a contest that is organized by Asosiasi Kopi Specialty Indonesia. And these are the coffee that were considered as a good representative from the lowest score of coffee quality, 80.25, up to the highest one, 86.75. So actually we have a very, very high score of specialty coffee. Higher than 85 is already a different grade, like specialty excellent something. So, and this is the type of coffee that we can see. Most of them have very good fragrance, aroma, flavor, and etc. So here we subjected the roasted beans from these 10 samples and then apply the extraction using a different kind of chemicals. So we use chloroform, methanol, and water with this ratio. And then we do derivatization using GCMS in order to analyze non-volatile metabolites. This is very important. We use this method as derivatization so that we can analyze non-volatile metabolites using GCMS. Usually GCMS is done for volatile analysis. Then we were able to annotate 97 metabolites. So among them, 46 is unknown. Then we use these 97 metabolites as an explanatory variable to construct a prediction model called orthogonal projection to latent structure, also OPLS. So this is a multivariate analysis method to find the relationship between Y and X variable. The X variable is this metabolite information, 97 metabolites. The Y variable is cupping score. So we use metabolite information to explain cupping score. So this OPLS introduces latent variable T that has a maximal correlation with Y. Then as a result, we have a regression model. So this T is then used to form a regression equation for Y. So I would like to give you an example of what kind of result we can generate from this kind of model. So this model also can give you the information on this VIP score, also called variable influence on projection. So in short, compounds of which VIP are greater than 1 are considered to be important for the model. In this case, compounds in which VIP are greater than 1 is considered to be important for cupping score. And regression coefficient, whether it's positive or negative, is a parameter which expresses the relation between Y and X variable. So it can be said that whether each compound contributes positively or negatively to coffee quality. So here we have a model performance and statistically important compounds. We make a model for each attribute. So for example, we have a model for fragrance, flavor, aftertaste, acidity, body, balance, overall, and final score. So we have many different models created for each of the score. And then surprisingly, we have a very interesting finding where almost all of the model, including the model for final score, says that galactinol, a kind of sugar alcohol, was proposed as an important metabolite that contributes positively to coffee cupping score. So this is proposed as a quality marker based on prediction models of 7 attributes and final score. It is also interesting that we found among all other metabolites that contribute positively to flavor, for example, flavor attribute, then we also have sucrose other than galactinol. So sucrose has been reported to have a high influence for the coffee quality, but the high correlation between galactinol and flavor of specialty coffee was proposed for the first time. So the next analysis is, after we know galactinol is important for coffee quality, is how we can increase galactinol concentration either in our coffee beans, or we can improve that in the way we process the coffee. For example, from fermentation, or we can increase the concentration of galactinol during the roasting method, or how do we brew the coffee. So this kind of information would be very useful for people who are doing coffee business. So the next, I want to show another example of how we can use metabolomics for coffee also. But this one, I want to say about coffee quality. So in 2011 or 2012, the world was really taken into storm by this world's most expensive coffee luwak. So naturally, coffee luwak is produced naturally. So in the wild, this civet is roamed free in the coffee plantation and then select the best and ripe coffee cherry, and then afterwards secrete the coffee beans with the excrete, and then the local people collect these coffee beans, wash them, sort them, and then dry, and then afterwards roast in very high temperature to make sure. So during this process, of course, it will be hygienic because everything will be dead at roasting temperature of higher than 200 degrees. Then afterwards, it results in coffee with exquisite taste. So this exotic production process and the rarity makes this kopi luwak the world's most priciest coffee. Then we found an authentic way to authenticate this kopi luwak, because it's very difficult to tell the difference between kopi luwak and not, because it depends on the coffee bean that is used as or the coffee cherry that is eaten by the luwak itself. So in 2013, we developed this method where we found the candidate marker that is only high in kopi luwak. So after digestion, these organic acids were present in higher concentration. We apply this discriminant marker to new batch of coffee samples that we bought in the market, and we were able to validate that only kopi luwak, authentic kopi luwak samples have this higher concentration of this marker. And for fun, we also test the instant coffee in sachet called Kopi Luwak, yang brand Kopi Luwak yang ada di sachet itu, the cheap one. And of course, the characteristic is very different with the real Kopi Luwak, the high quality coffee. And then this is the bar graph where you could see that the concentration of the markers that we propose, citric acid, malic acid, and the ratio of inositol pyroglutamic acid, this multi-marker profiling was able to validate the applicability of the discriminant marker. And that was my first research on the use of metabolomics for Indonesian bioproduct. And the research was also validated by feeding the coffee fruit to luwak. And then we confirmed that the markers were indeed increased after digestion. So this was very well reported in the many mass media, also in the TV, local TV in Japan. We were interviewed by BBC UK. It was in NPR, even in Science magazine. And that opened a lot of doors and was the start of many different kind of collaboration with different institutes in Indonesia. So I don't stop there, because kopi luwak is just one kind of coffee that Indonesia have, but we have so many rich diversity of natural coffee that is already high quality. So I have already finished the research on mapping of all Indonesian specialty coffee. In this case, I analyzed 9 high quality coffee, the specialty grade coffee from all over Indonesia, and it was published in 2019. So as Indonesian people, we have to know what is the characteristic of Bali coffee. Not only descriptive, but we know what kind of marker is high only in Bali coffee, in the coffee from Western Indonesia, from Eastern Indonesia, from Papua. Then we could preserve this biodiversity and then continue this information throughout our many generations for our young generations later on. So I did the same also for tempeh, because tempeh is our national food, and I'm very proud that we are the nation of tempeh. So, but of course, maybe we are never aware of what makes tempeh from different region different. A lot of research have already dealt with the microbial consortia differences between this tempeh and this tempeh, but no study focus on the differences in the composition or metabolite profile of tempeh that is produced traditionally, for example, by tempeh crafters, which makes maybe 90% of the whole production nationwide that is produced by small home industry. What is the difference between the tempeh that is produced in a semi-modern way and the one that is produced in modern way, for example, like already with ISO 9001, for example. And no one also knows the difference between tempe produced in Bandung, in Malang, Purwokerto, Surabaya, Yogyakarta. So our student, in fact, went to all of these locations. So she left from Bandung, from ITB, going to Surabaya first by plane, collect the samples from Surabaya and Malang, and the Surabaya and Malang tempe was produced in the same day, was delivered by ojek driver, and the cost of one tempe sample was, one location of tempe was about 500,000 rupiah. And that is not about the tempe, it's about the shipping from Malang to the airport and Surabaya to the airport. And then she moved to the same day from Surabaya to Yogyakarta, also collect the tempe samples produced in Yogyakarta and also Purwokerto on the same day, and then go back to Bandung where she collected the samples that were produced in Bandung and Bogor on the same day. That's why we could discriminate what is the difference between tempe produced from all of these regions at once. Then it's very interesting to see that there are distinct differences between tempe produced in West Java and East Java. I have to mention also that each dot represents the tempe produced from different makers. It's not from one market. So even it's produced in 3 different locations, 3 different markers, Bogor tempe has its own characteristic and it's very close to Bandung tempe. So here you could see the difference between what kind of differences are there also for tempe produced in a modern way, semi-modern, and traditional. So finally, I would like to explain also how metabolomics can be used to predict complicated phenotype, for example, such as fruit ripening. So we know that mangosteen or many different kind of tropical fruits like banana, mango, pineapple, 4 of the important fruits from Indonesia I choose to analyze. The first one is mangosteen, because mangosteen is very difficult to keep for a long time because it automatically ripes to the following stage for 2 days in room temperature. So we tried different methods to prolong the shelf life of mangosteen, and only low temperature at 14 degrees worked. Then we tried to understand under 14 degrees what changes in the metabolite profile. So here you could see that in the normal ripening in room temperature, fructose, glucuronate, galactose, acid increase over time from left to right in the red, as shown in the red mark. But in the case of 14 degrees or 12 degrees Celsius, then the progression or the increase of this metabolite stopped or becomes much slower. So this is what happens when you inhibit ripening. So if we can find a method to mimic or modulate the effect on to inhibit the progression of these metabolites, then we can prolong the mangosteen shelf life longer. So similar concept we are already doing with pineapple. Now we can make pineapple stay longer or shelf life longer. So this is the latest publication. Similarly with pineapple. So and I would like to close today's talk by saying that metabolomics is very, very useful for discriminative purpose, for informative purpose, as well as for prediction purpose. So I don't have a lot of time to explain a lot about my other research topic, but I hope that you would stay and attend 4 of my students' presentation today. And they will also talk about many different ways that metabolomics can support post-harvest development strategy, disease control in agriculture and aquaculture products, as well as quality improvement. So all of these things are very, very important to strengthening nation's food security. Especially, I could not stress enough the importance of making our food security very strong. In times of this kind of pandemic, we have only our nation food security to rely on. In this pandemic situation. Most importantly, we have to pay attention on this issue. So if you would like to see our research team presentation today at 2:40 to 3:20 p.m., research about tempe metabolomics will be delivered by my PhD student, Adinda Kadar, and 3 of my Japanese students will give a talk on cacao metabolomics, shrimp metabolomics, and also fermented beverage metabolomics. So with that, I would like to thank all of our collaborators from Osaka University, Kyushu University, ITB, Indonesian Coffee Cocoa Research Institute, ITB, and ITB University. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Dr. Sastia for the interested informations and presentations from you. So we know that very important of the metabolomic related about how we can conserve about the biodiversity in the world. And then we also know about so many functions of the metabolomics in our life. And then after that, maybe in a chat room or maybe in in a chat room, some of the questions pointed to Mr. Wali Pansikal and also to Dr. Sastia, and then we try maybe to read some of the questions we have already chosen to answer by both of our keynote speaker. Okay, because now we are entering for the session of the questions and answer, and then for the first questions come from Mr. Ahmad Junaidi, and then this question pointed to the Mr. Wali Pansikal. Okay, we try to read the questions. Mr. Wali, have you still here? Hello, Mr. Wali.
I think you are still muted.
Okay, I'm here.
Okay, well, the questions, number 1 for you, we will try to read. Based on your experience, Mr. Wali Pansikal, what is the biggest problem in biodiversity conservation? And in my opinion, for the developing country, cost is one of the constraints. Thank you.
Well, I don't know if there's one single issue. I like to think of it as the mnemonic device HIPPO, HIPPO. So habitat destruction, introduced species, pollution, population, and overconsumption. So HIPPO. I think it's a combination of those 5 things, and then we can certainly add climate change to it now. I don't know as one, it depends on the location, the region, what species we're dealing with, which one of those actually 6 things affects the biodiversity the most. So it's a combination of things, or some ones are more prevalent than the other ones in different locations. That's the best answer I have for that.
Okay, thank you. So it means that there are so many factors, maybe effect for the biodiversity in the world. It's different to the area or maybe some kind of that. Okay, well, and then we also still have the second questions from our students from University of Bengkulu, from Nadia Rosianti. She's one of the biology student. So she want to ask to Mr. Wali Pansikal. We know that each year Idea Wild distribute over more than 400,000 worth of equipment, including digital cameras, spotting scopes, mist nets, stands, binoculars, and GPS units to over 400 conservation projects worldwide. So how do we get the small grant for research tools from Idea Wilds, and how long it takes to propose for the proposal?
Well, like we explained at the end of that presentation, you need to go to our website, and then the application form is on the website, and the guidelines are on the website. You can apply anytime during the year, and right now, if you apply in month number 1, within 2 months, we will answer your yes or no. So if you applied in November, we would answer your question in November, October, in January. So after you apply, we will tell you yes or no. We've been able to shorten the period because of the pandemic, because we actually took 3 months off. We did not accept any proposals. And then in July, we began accepting proposals again. And so we got rid of a backlog of projects. And it looks like Anne-Marie, our project director, has put on the chat the website for Idea Wild, and that's where you will find the application form and the guidelines.
Okay.
2 months after you apply, you will get an answer.
Okay, so is it open for all? I mean, for all of the common people, it's not only included
Yes.
for students later also, and some
We
degree
have
researcher.
lots of bachelor's degree students that apply, mostly masters and PhDs that apply, and some postdocs, and then some people involved with agencies, and some people involved with nonprofit organizations. So it's basically any people that are interested in conserving biodiversity and are passionate about conserving biodiversity at any level in their career.
I think it's very interesting. So we also can ask our student to propose the proposal
Yes.
to follow for this grant too.
Yes. Most of our proposals we accept are from master's and PhD students. Yes.
Thank you. And then we go to the next questions. It's still from the students of biology from University of Bengkulu. His name is Alfred Ginting, and he would like to ask to Mr. Wali Pansikal. So this is the question. Maybe you have heard about the Komodo National Park on the Indonesian Komodo Island. The Indonesian government wants to create a Komodo Jurassic Park project, and this project is currently underway. But this has generated a lot of debate. Based on your experience in the field of conservation, what do you think about this? Will this Jurassic project disturb or maybe destroy the habitat of Komodo dragons or make the life of the Komodo dragons better? Thank you so much.
I have to explain that I have no idea about this project, and I don't have any idea what this project entails, the planning or anything. I assume that if a bunch of really smart conservation biologists were involved in the planning, you could probably have both a tourist attraction and conserve the Komodo dragon. But if it's just all about creating and destroying habitat, I don't think that's probably a good idea. But I'll have to admit, I don't know anything about this project.
Okay, thank you so much. And then we try to go to the other questions to Dr. Sastia, pointed to Dr. Sastia. So we try to read for the first question. Dr. Sastia, when we want to try, for example, like organoleptic for the copy, how many panelists that you use and replicate in the analysis, minimal use of the panelists, and also the analysis of OPL to analyze in the biodiversity species like animal or plants?
Okay, thank you very much. It's a very nice question. Actually, the number of panelists depends also on the quality of panelists. If you use untrained panelists, means like anyone without training will just be subjected to analysis, you need to have a very high number. For example, more than 70 panelists is required. If you have a semi-trained panelist, about 20 to 30 semi-trained panelists, which means that they are aware of what kind of attributes and how to conduct the sensory analysis. If you use trained panelists, then sometimes, ideally, we need 5 to 10. But in our case, we tried to use trained panelists, 5 trained panelists, but the result from one panelist that is like a maestro, for example, like very, very skilled maestro, is actually much better to create a model rather than 5 trained panelists. So in that paper, we use one licensed Q grader, which is Pak Yusianto. This is the coffee maestro of Indonesia, and only one trained panelist is enough. So that is, it truly depends because, for example, if he can predict, I mean, his consistency is very, very good. It's better to just use one compared to 5, and then it will make a very big deviation among panelists. So it really depends. Either you have a very large number or a small number but high quality of trained panelists.
Thank you. And then, okay, for the second questions, maybe these questions in Bahasa Indonesia, so we would like to translate. So, Dr. Sastia, maybe can we use any other kind of the seed, maybe or bean, to produce of the fermented tempeh? I mean, because commonly we use of the soya or soybean, But maybe how about the change? If we change the sheet or maybe the bin by another of kind of plant? Thank you.
Yes, it's a very interesting question because it's actually a topic of one of our PhD students. So she is about to submit her first paper where we produce tempe from 5 different legumes. For example, mung bean, kacang hijau, kacang merah, kacang koro, dibandingkan and we compare that with soybean. Then we compare the differences among them using metabolomics. So yeah, we have already done that kind of research, and it would be interesting to expand. Mungkin kita bisa produksi lebih banyak tempe dengan menggunakan lebih beragam jenis legum dan membandingkan which one gives the best quality of tempe. So yes, sorry.
So we try to make tempeh by variations of the bean, like peanut, and then like red bean and the other one.
Yeah, so it's very, very interesting, very similar. So our students found that, for example, to increase the quality of tempeh, in terms of the nutritional quality, we can make a more nutritious tempeh that has higher amino acid content by combining the legumes, not only using soybean, but soybean with what kind of legume. Then when it's combined, the nutritional quality, for example, the amino acid composition becomes higher and also becomes more lebih banyak. It becomes more diverse. So we do that kind of research also to improve nutritional quality of tempeh.
Okay, thank you so much for a very nice answer, Dr. Sastia. And then, okay, we also have any other questions to Dr. Sastia too. How about how we can maximize the metabolite that could be detected, and then what kind of the preparations that we have to prepare?
Okay, so the question comes from a good friend of mine, Ibu Marlin. Apa kabar Ibu Marlin? So about the maximizing the metabolite for coverage, it really requires extensive optimization on what kind of solvent used for the different particular sample. Let me just give you an example on mangosteen. So mangosteen, depending on what type of mangosteen, for example, fruit part or flower part, or so in the plant part, if you analyze fruit or you analyze the root or you analyze the stem, it will be different, the characteristic. And similarly, depending on the sample that you use, if it's semi-solid, liquid, or solid, also you have to optimize each one. And we cannot use the same method universally. Usually we start with our universal method that we describe, but we have to optimize for different kind of samples and to maximize the metabolite coverage. Also, the characteristic of each sample is different. For example, like tempeh, if we just do the extraction directly without, for example, lyophilizing it first, without freeze drying, a tempeh becomes very sticky. And then afterwards, we cannot extract properly. It decreases the efficiency of extraction. And if we want to analyze, for example, coffee beans, so coffee beans usually we grind first, right? So very simple thing like how fine is the grinding? What is the size of the particle? You also have to pay attention because if you just crush randomly, you will have different size of the particles of the coffee bean that you grind. Jadi bubuknya itu halus atau tidaknya, it will determine the efficiency of your extraction method. So if you don't do it in the same way, then you will have a very fluctuating result. So you have to understand the characteristic of each sample. And also, the method optimization is also important. For example, like we analyze udang, shrimp. And shrimp has a lot of protein. And then if we don't remove this protein from the sample, then it will damage the machine, for example. So we have to do deproteinization before we put it in the machine. Otherwise, for example, the column or the mass spec will be damaged.
Okay.
So how do you prepare the sample that cannot be stored for a long time? So every single time you analyze the sample, usually we have to deeply do deep freezing using liquid nitrogen at minus 80 degrees. So that is the first step to preserve the sample and stop metabolism. So at minus 80 degrees, all metabolic activity will be stopped, enzyme activity. Then the sample can be stored longer. Then you have to do the extraction soon after. After you do the extraction, you have to freeze dry. Then you will be left only with lyophilized extract. Then this lyophilized extract, please keep in minus 30 at least, minus 20 or minus 30 deep freezer. I know in Indonesia it's sometimes difficult because sometimes ada mati lampu ya, but,
Yes.
but that is the best way to keep the sample longer. Immediately do the liquid extraction quenching, extract the samples, lyophilize the sample, and keep it under minus 30 to keep the quality. Don't keep the sample as an extract, liquid extract, never do that. And also don't keep the sample just as it is because it will be damaged. The damaged tissue or anything will be damaged, even keeping it at minus 30 before extraction. There is
Okay.
some technical tips. Yeah.
Thank you so much. And then the other questions also pointed to you. The questions is about, based on your research, we found that the galactinol is one of the important compound in the coffee. So do you think it is gonna be the standard for good quality coffee in Indonesia?
I hope it can be a gold standard for quality of coffee in the world for specialty coffee because it's the first quality marker. So I'm very excited. The paper is not yet published, so I don't know the response of the reviewer. I could say for sure after it's published and it undergoes peer review.
Okay, and then the other questions also related about the metabolomic research about food. So do you think that you want to apply your result in public to improve the quality of the product? For example, like a food product, maybe.
Yes, actually, I wanted to give an explanation about how we use the second fermentation. So the story about kopi luwak, we isolate a collaboration with Professor Pinkan, who is also going to give a talk today in the afternoon as an invited speaker. She's actually my teacher and mentor. So Professor Pinkan and I collaborate, and then we isolate bacterial isolates from the luwak excrement. Then we want to produce coffee without involving animals. So maybe Wally Pantikal, you agree that it's probably best not to involve animal in the production process of coffee. So we have done that in vitro. So we just take the bacterial isolates and then we use these bacterial isolates for fermentation, put it in the coffee bean, and then it could increase the cupping score by two degrees. So it's very, very high effect. And we have already published that last year. And we have registered a patent in Indonesia for the improvement of coffee quality through second fermentation process.
Okay.
So yeah, so it's possible. But the most important thing that we did is we don't do it in random. We do it in a targeted way in which the process was optimized by monitoring the metabolite marker composition. So we know what we are after. So we don't do just a random optimization, but we try to find the best bacterial isolate and the best fermentation condition in which the markers that we target can be increased. So the concentration of the markers that we know is important for coffee quality can be enhanced. Then during that way, through that way, we could significantly increase the coffee quality in a targeted and a systematic way.
Yes, thank you so much. I think actually there are some of the several questions maybe entering to the chat room, but because we have a limit times to finish for our presentations, maybe before we finish for our presentations, we would like to conclude some of the presentations that have been presented by our board of keynote speaker, and we would like to maybe to conclude some of the conclusions from the presentations of Dr. Sastia. We do know that the metabolism is one of the very important things in the world, especially for nowadays, because we know that the applications of the metabolomics we can find in any in many cases. I mean, in many aspects in our life. So, like maybe you have told that in a bioscience, food agriculture, and especially for some of the product and the bio industry and medical and many approach. So I think we know that what you have done is also very important, especially to conserve about our biodiversity. But I mean, the perspective maybe are rather different with what the Wali Pansical does. Maybe in a laboratory scale, but I think it has sense of the related and strengthen each other. I mean, because it's also very important to keep for our biodiversity. So I think that's a very important, especially for this subject, right?
So today, this plenary session has covering this first part, but both are at the same theme, that is biodiversity. The speaker first emphasized on the field of biodiversity, that is life biodiversity in the field, in the jungle, in the forest, Rocky Mountain, and so forth. And the second speaker is emphasizing on the laboratory, but finding the meaning of diversity and the marker diversity to understand, and both are strengthening each other to support the conservation issue. I think that is all.
Yeah.
Yeah. So we come to the close session.
Okay. We finished for our presentations. So we would like to give the certificate appreciation to our distinguished keynote speaker. For the first certificate will be given to Mr. Wali Pansical. So cannot open. Okay.
I'm not sure what. Oh, thank you very much. Thank you very much. I very much an honor to be there. Very much an honor to speak at your conference. I very much appreciate what all of you are doing to make this planet a better place. Thank you.
So this is the certificate for you, Mr. Wali. And then the other certificate is going to
Sastia.
yes, Dr. Sastia.
Yeah.
I wish that maybe Mr. Wali, if the pandemic has end, maybe if you have any time, you can visit to Bengkulu province, because you know that Bengkulu province is one of the mega biodiversity province in Indonesia, because we have a lot of the mega biodiversity. Maybe you can visit someday to Indonesia, especially for our province Bengkulu.
Yeah.
I will take you up on that offer. Thank you for the invitation.
Thank you. Thank you. Again, before closing, my name is Fairul Muslim,
And
my
me,
friend
Rizky Hadiwibowo.
would like to appreciate both of you, Mr. Walter Dean and also Ibu Sastia, who has already speak to us about the biodiversity in the different side of view. And then before closing, we would like to remind all the participants that this after occasion there is still a lot of session. That is, in the next session will be at
11 o'clock.
At 11 o'clock.
Yes.
That is invited speaker, and then there will be parallel session, invited speaker session, and then parallel session from let's say from 10:30 to 11 p.m. and then from 11 p.m. to 1 p.m. parallel session 1, and so forth until the closing ceremony that will be done, that will be occurs at 4 o'clock p.m. Okay.
So once again, we would like to say thank you so much to Mr. Wali and Dr. Sastia, especially for Mr. Wali. Good night there, because in USA now has been night. And also to Dr. Sastia, maybe we will contact you continue, because we also want to continue about health reasons probably maybe happen in University of Bengkulu and especially.
Okay, yeah, I would love to collaborate with the audience here today. Thank you very much.
Thank you so much.
Kingston.
Thank you so much, Mr. Wali.
Thank you. That is all, our dear audience, ladies and gentlemen. As we have finished in this plenary session, we thank you for all of you. And Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Okay, we would like to remind for the presenter, especially for the international seminar now, because we divided become some rooms. For example, like an for every single scopes, there are some sub-rooms because we have only one single invited speaker for each scope. So maybe for the first time, you have to join to the room that's selected to the invited speaker. So it's mean, for example, like the scopes of the biotechnology and also the molecular biology, you can join in the room 1. And then the other scope, maybe you can join in the room 3, room 5, and also room 7. So after that, maybe after we finish the presentations from the invited speaker, maybe you can come back to your own parallel sessions rooms. Okay, maybe we try to remind that. Thank you so much.
Saat ini, kita berada dalam masa sulit. Terbatas dalam gerak, terpaksa oleh jarak. Menjauh dari dunia yang sesak. Yang tak lama, tak sejenak. Saatnya berjeda. Menjaga jarak, satu-satunya solusi. Ada kalanya kita perlu berhenti agar semakin cepat pulih kembali. Perhentian ini pun menjadi sebuah momen untuk kita merefleksikan diri. Waktunya untuk peduli, memberi ruang untuk dunia sedikit beristirahat. Banyak rindu yang harus ditahan, banyak rencana yang harus ditunda. Sebarkan harapan untuk keselamatan bersama. Kita semua saling menguatkan. Ini adalah momen untuk kita mencari sudut pandang yang tak pernah kita temukan sebelumnya, menjadikannya sebagai energi untuk nanti bergerak kembali. Ini adalah momen untuk semua menyatukan asa dan harapan. Dan ketika hari itu tiba, semua ini akan membaik. Manusianya, begitupun semestanya.